You're in the third row. That's better than anybody else, but welcome all of you. We're very happy to see you here for this edition of the Della Pietra lecture series. The Della Pietra's belief and our belief is that by bringing scientists who are doing exciting, innovative, influential work, like today's speaker, bring those scientists here to talk to groups of high school students like this one, will help convey the enthusiasm of doing science, the joy of doing science, and will help nurture in at least some of you a desire to continue along the science path. Our country needs a lot of scientists, and maybe some of you out there will become the next generation. Science needs good problems, questions, puzzles, goals, but it also needs innovative, creative people to work toward those goals, to try to solve the puzzles, and the most creative people are always, sad for me to say, the young ones. So that's where the future breakthroughs in science will come. They'll come from younger people, people like you in a few years. And I told Stephen that one of his goals today was to produce at least 2 PhD scientists out of this group. And he said, well, he hopes he'll get 2 to come to Stanford. So our speaker today is Professor Stephen Quake. He received his bachelor's in physics and master's in mathematics from Stanford University, and got a PhD from Oxford University, where he was a Marshall Scholar. After a stint at Caltech, working, among other things, in Stephen Chu's lab, the former Secretary of Energy and Nobel Prize winner in physics, he moved to Stanford, where he started the Department of Bioengineering, and where he is the Lee Otterson Professor of Bioengineering and Physics. He has many prestigious awards to his credit. He's a member of the National Academy of Sciences, the National Academy of Engineering, and the Institute of Medicine. Well, that's the trifecta right there of national academies. He's won many prestigious international prizes, including the Sackler Prize in Biophysics. He's known for his pioneering new approaches to biological measurement, especially to microfluidics. He made the first single molecule DNA, and he was one of the very first people in the world to see his own genome. I asked him if that caused him to change his view of himself, and he said, well, you know, the National Institutes of Health gives lots of money for bad things, finding out genes that cause bad things, but not much for finding genes that cause good things. So he only learned bad things about himself, no good things. But that did cause him to change his behavior a little bit. The title of his talk today is Precision Measurement in Biology. Let's welcome Stephen Quake.
Thanks so much. So it's a pleasure to be here today. They didn't tell me a lot about you, so I want to get a little data before we jump into it. How many of you are seniors? Most of you. All right. Juniors? All right. Sophomores? All right. Freshmen? How many of you want to go to Stanford? Come on, get them up. Let's go. How many of you are scared an earthquake's going to drop Stanford in the ocean when you go there? That's what they told me, yeah. I grew up not too far from here. That's what they told me. They also told me I missed the winters. It's all a bunch of BS, all right? I'll tell you what, in 20 years, I've never missed a New England winter. If you have any questions during the talk, raise your hands. Let's keep it sort of informal, and I'm going to try and gear it to where I think you're going to enjoy it, but if not, let me know. How many of you have taken physics? All right, good. Biology? Oh, good. You know what DNA is? Yeah, all right, we can skip that slide then. I got my start as a physicist, as you heard, and now I'm primarily working in biology and genetics. And the talk is sort of geared, because we're in a physics center here, physics and math center, to and physicists, they tend to be a little ignorant of biology, so I try to help them learn by analogy. And so I'm going to start off with some ideas from physics, including this idea of precision measurement, which dates back 300 years in physics. More or less originates with this guy. Anyone know who he is? Dead white guy. No? Any guesses? Yeah, all right, that's pretty good. All right, I'm impressed. If you Google Galileo, what you find out is he said this wonderful thing: measure what is measurable and make measurable what is not so. And so the first lesson I want you to remember from this talk is do not believe what you read on the internet. Because as far as I can tell, he never said that. No one's been able to document it, although it says on the internet all over the place that he said that. Even though he probably didn't say it, he should have said it, because it was really what his sort of science was about. He really thought carefully about how to measure things and, really set the course in physics for 3 centuries. And one of the things he's very famous for is building the most precise telescopes of his time and turning them to the heavens and making all kinds of astronomical discoveries, including the fact that the moon is not a perfect sphere, and there's all kinds of things on the surface. But he also, and of course, got in that famous fight with the Catholic Church about the heliocentric universe. But he also did many things besides astronomy, and was involved in making measurements in mechanics and motion, and kind of made some discoveries that were precursors to Newton's law. He figured out that you're rolling balls down planes. They go as the distance goes, like the square of the time, and I kind of worked that out phenomenologically. And he measured time by trying to sing and keeping track of the musical rhythm to do his timing, and was really sort of a phenomenal person, and very much revered by physicists. And in fact, if you're ever in Italy, you can go to Florence. You can go to the Science Museum there and see not only some of his instruments and his books, but his finger has been preserved as an artifact to come pay homage to. I went and saw it last summer. It really, really quite impressive. They got a tooth too. It's really quite moving and pretty interesting. And his ideas about precision measurement, as I mentioned, sort of really influenced the field. And you can pick a few examples from the 20th century as well. I mean, just thinking about spectroscopy of the simplest element, hydrogen, and ever more careful measurements of it have led to the development of amazing technologies, and forced us also and led to discoveries that have forced us to revise our pictures of the physical universe over and over again. Here's some folks who are deeply involved in that: Stern, Rabi, Lamb. Each of them, by doing more and more precise measurements of hydrogen, have made phenomenal discoveries. And in fact, there's a great book that I recommend if you're interested in that. The Story of Hydrogen. It's kind of a history of, I think it's just called Hydrogen. It's by a guy named Rigdon. And it tells the story of the element and all the amazing sort of discoveries about the universe that have come out of just trying to study the simplest element. And from these folks, for example, the techniques they invented to study hydrogen not only led to great scientific discoveries, but also great technological advances. So Rabi invented NMR, which is used today for the GPS system, for MRI imaging, and so forth. And so the story of precision measurement is this kind of intertwined story of people trying to measure things more carefully, to advance the frontiers of science, inventing new techniques to do that, which then lead to amazing technological discoveries that are well beyond what they sort of originally intended. And that's been a great story in physics. Hasn't really been the story in biology, but biology is changing, and we're kind of in this amazing period right now where there are great new measurement techniques that are really changing biology. One of them is around DNA sequencing. The ability to read out the genetic code in a person's genome or an individual's genome has grown amazingly over the past decade. And I'm going to give you some examples of how we've been using those techniques in my lab to do precision measurements in biology and medicine. And another technology that we've been deeply involved in has been the development of microfluidic tools, which you can think about as the sort of biological equivalent of the integrated circuit, which making chips that instead of having transistors and wires have valves and pumps and pipes and so forth. And they've become a powerful tool for biological automation. So you guys told me you know what DNA is, right? It's It's the molecule that is in all of our cells, sort of the blueprint for making the cell in the organism. And it's a repository of information. The information is encoded chemically. So the structure of the molecule is this famous double helix. There's a great story around that. You read Jim Watson's memoirs. And there's sort of a four-letter alphabet, ACTG, which refers to the four nucleotides, and they pair up in a certain way, and the information is stored redundantly because of the way they match. But the sort of take-home lesson is you can use chemical molecules to write in instructions for making proteins in DNA. And so the question is, can you read that out? There have been a number of techniques developed to read out the chemical information in DNA, dating back decades. But it's only in the last sort of 10 years that we've really developed ways to do that at a whole genome scale. And I'm going to tell you about a couple of them. But before I do that, let me just give you a few numbers about DNA. So if you took the genome in your cell and stretched it out, all the DNA, laid all the chromosomes end to end from one cell, it'd be a couple meters long. So that's a big molecule, right? You got 20 odd chromosomes, 46. You lay them out and get 2 meters. So that's huge molecules. That's in every single one of your cells. And you've got about 100 trillion cells. And so if you took all the DNA in all of your cells, you'd go back and forth from the Earth to the sun several hundred times. It's really quite astonishing. And the way that works, of course, is that although the molecule is very long, it's very skinny as well. So it's got atomic dimensions along two of its axes, just a couple of angstroms wide. So I'd mentioned that this is an amazing time in DNA sequencing. This is a figure from Nature magazine's 10th anniversary of the Human Genome issue. And this is time on this axis in years from 2000 to 2010. And there's a bunch of things being plotted on this axis. One of them is the cost for sequencing a megabase of DNA. And you can see that that's dropping exponentially with time. Okay, it's falling off by factors of 10 as the years go by, and that's due to kind of the feverish technological efforts to develop new measurement techniques in genome sequencing. The other thing here shows sort of the amount of genetic information that's been measured increasing also exponentially over time. The number of genomes have been growing incredibly rapidly. And that's been great. You've probably read in the papers about this and this genome being sequenced over time. And it's been sort of an amazing time in science. But you've also got to recognize that there are skeptics and folks saying, what does all this genome sequencing got us at the end of the day? This is an article that Nick Wade wrote in the New York Times a couple of years ago where he said, this genome sequence hasn't really given what you promised us. We spent $3 billion of taxpayer money doing it, and you promised that you'd find the roots of a bunch of common diseases like cancer and Alzheimer's disease and cure them. But that hasn't happened yet. In some ways it's a fair criticism, but in other ways it's not fair because many of the great discoveries of the genome were things that weren't anticipated when these folks were writing their proposal. And this is sort of another interesting aspect of how science works. When you sit down, you write your proposal to the government, you say, this is what I want to do. I think we'll find something interesting. It may be this, but sometimes it turns out to be that instead. And you make some discovery, it leads you in a completely different direction. And that's kind of the pleasure of doing science and doing that. And so while Wade is right that a lot of these common diseases have been very difficult to unlock, even with genomic techniques, there have been many other things that come out of the human genome that have been incredibly useful for human health and for science. And I'll give you a few examples of that. John mentioned in my group that we'd been involved in developing single molecule sequencing and then sequenced my genome with it. And I'm not going to tell you that story today, but it was a lot of fun. We can, if there's time for questions at the end, we can talk about that. I want to tell you about a few other things we've been doing that are sort of getting off the beaten path a little bit. Everyone started thinking, well, let's just sequence human genomes. And we began to think about other ways to use these instruments to do precision measurements as molecular counters and as other ways to take sort of snapshots of different biological ecosystems or of human health. So here's one example. Humans are diploid. You're supposed to have 2 copies of each chromosome. You inherit one from your mom, one from your dad. So we're distinct from, say, most bacteria in that sense, who have only 1 copy of their genome. We've got 2. And you can prove that you're human and you're diploid by doing what's called a karyotype, where you take cells, break them open, and lay the chromosomes down and take pictures of them under a microscope. This is a human karyotype right here. And the chromosomes have all been, there's been quite a bit of, they don't tell you this in the textbooks, but when you do a karyotype, it never looks like that. The chromosomes are all randomly put across a microscope slide. So someone then does a bunch of cut and paste in Photoshop and lays them all next to each other so you can see what the sizes are and whatnot. And so they don't really end up looking like this until you've done some artistic editing. But there should be 2 copies of each chromosome. They've been stained with these bands, so that means something that people looks at these things. 2 copies of 1, 2, 3, 4, 5, all the way down. See, this person right here has 3 copies of chromosome 21 though. Now, that's not good. It's called a trisomy, or an aneuploidy more generally, if you don't have the right number of chromosomes. And trisomy 21 is more commonly known as Down syndrome. Right? And that's sort of the most common genetic disorder for live births. And and these sorts of aneuploidies, where you have the wrong number of chromosomes, affect close to 1% of live births. So it's a pretty significant issue. And doctors recommend pregnant women get tested for this. And it shouldn't be such a hard measurement. All you have to do is count to 3, and most doctors can do that. But the challenge comes in actually where you get the material from. How do you get the baby's chromosomes before the baby's born? What they end up doing is taking a big needle and sticking it right in a mom's belly and right next to the fetus and trying to capture a few fetal cells. And they take them out and grow them up and try and do that karyotyping that I showed you. And there's a couple ways to do this. Amniocentesis is the most famous one. There's another one people do now called chorionic villus sampling. As you can imagine, this creates risk both for the mom and the baby. And I first got interested in this problem when I became a parent and saw this done to my wife and unborn daughter, and then this done to my wife and unborn son. And I realized this is not a great situation where you're having to take a pretty significant risk in order to get some information. And I became interested in this question of how do you come up with a non-invasive way to measure genetics. And that rattled around in the back of my head for a long time. And one day I was sitting in my office flipping through an issue of Science, and I stumbled across a reference to this very old literature, which dates back to the 1940s. These 2 Frenchmen here discovered that there's a certain amount of DNA that just floats around in your blood. Okay? You normally think about DNA, it's got this privileged position. It's the blueprint for the organism. It sits there in the nucleus giving out the instructions for what to make. And that's true, it does all that. But it turns out there's naked DNA molecules that float around in your blood. And those are kind of garbage. It's sort of the garbage from dead cells. When a cell dies, your cells are dying all the time as well as dividing. When a cell dies, breaks open, nucleus breaks open, the genome gets spilled out in the blood, it gets chewed up into little pieces, and those little pieces are just there in the blood. And so if you take a blood sample and separate out all the cells from what's left, that stuff that's what's left, there's a bunch of DNA there. And these guys discovered that. A long time ago, the paper was forgotten for quite some time. Eventually rediscovered. There was a funny little field that worked on this for a while. This is their data from the paper. They looked at a bunch of different people, normal, different afflictions. One affliction they looked at was pregnancy right here. And they noted, this is the amount of sort of DNA and RNA, they noted that in this case of pregnancy, the values seemed a bit higher. So they kind of knew that something was going on in pregnant women with this DNA in the blood. And what turns out at the end of the day is that some of the DNA in the blood of a pregnant woman is from the baby, because the fetus has got cells that are dying, and there's some of that in her blood. And so by taking a little bit of blood from a mom's arm, which is very low risk and considered non-invasive in this business, you can get access to some of the baby's DNA. The challenge here is that it's not as simple as counting to 3, because most of the DNA in the mom's blood comes from the mom's cells, and only a few % of it comes from the baby's. So the mom, who's in most cases normal, is putting in 2 copies of every chromosome. The baby, if it's got Down syndrome, is putting in 3 copies of chromosome 21 for every 2 copies of chromosome 1, every other chromosome. But that creates a very small difference. And so the challenge is, how do you measure that small difference? And what we ended up doing was using these sequencers to do that. So my student, Christina Fan here, went and purified cell-free DNA from the blood of pregnant women and put it on a sequencer, sequenced several million molecules from each sample. Each of those molecules took the sequence, mapped it back to the reference human genome. So here we're using the Fruits of the Human Genome Project, and asked which chromosome did that molecule come from. So every molecule sort of votes for a chromosome, and in doing so, you can look for overrepresentation of a chromosome, even if it's only by a few percent. Turns out in biology, that's a pretty tough thing to do. It's hard to measure things with precision of a few percent, but this ends up being these technologies that let you do that. And so we look for overrepresentation, or every molecule votes for a chromosome, and we're looking for voter fraud because someone's voting more frequently than they should be. And this is what we see for chromosome 21. This is the relative representation of the molecules from chromosome 21 out of the sequencer. The red data points were all from blood samples that were taken from women who were pregnant with babies with Down syndrome. The blue samples were from women whose babies did not have Down syndrome. And you can see it really clearly separates. Chromosome 21 is overrepresented there. You can sort of draw a line that separates them. This is Yair Blumenfeld here, who was our clinical collaborator, helped us get all those women to join the study at Stanford. Now, there's nothing special about chromosome 21 as far as the measurement goes, and in fact, we're able to look for aneuploidy or trisomy of any chromosome. And this is all the chromosomes right here in a funny order. And this is their representation here. So this is chromosome 21 right there. This is the data I showed you, a bunch of the red ones separated here. It turns out we had 2 women in the study who were pregnant with babies with trisomy 18. If you go to chromosome 18 right there, see those 2 black outliers, right? Those are the 2 women whose babies had trisomy 18. And we had 1 woman in the study who was pregnant with a baby with trisomy 13, that outlier right there. So you can read off, these 3 are the major chromosomal abnormalities that happen. 21 is the most common, and then 18 and 13 are about an order of magnitude less common, and very little else is survivable. So we felt very happy about that. We'd solved this long-standing problem of how you develop a non-invasive diagnostic technique for doing prenatal diagnosis. We published the paper in 2008, and it set off an enormous amount of excitement in the field. A bunch of clinical trials were launched right away, both in companies and in academic centers, and they reported out very quickly in 2011, 2012. So just 3 or 4 years later, they all published, and on thousands of women they looked at, they tested this technique. This simple counting principle we'd invented worked exactly like we said it would, and it's been in the clinic now for a little more than a year. This year in 2013, something like 500,000 women worldwide received the test. You can sort of do the math. The fatality rate of the conventional technique is on the order of 0.5% or so, and so that's several thousand lives that have been saved by people avoiding doing invasive diagnostics.
Question about that.
Yeah.
Do they completely rely only on this test, or when your test suggests there's a problem, do they then go in and take some antibiotic?
Yeah, that's a good question. So it's, the question, has it completely replaced amniocentesis yet? And the answer is no. So the way they're using the test now is they give the test. If the woman comes back positive, then they send for an invasive follow-up. But because the rates of the occurrence of trisomy are so low, you're effectively still saving 99% of the people from having to have an invasive test. So,
and what percentage of the follow-up tests give back positive?
Oh, the concordant, well, it's interesting. So let me, before I answer that, let me just mention, so I was involved in a company that was commercializing this. They were acquired earlier this year by Illumina, and there's about 5 companies that are offering versions of this test right now, and they're doing it to different degrees of let's say, quality. Some of them are creating a very high quality version. Some are doing it more as a screen, so it's kind of has a lower performance. And so I can say for the companies that are at the high end of the spectrum that are doing the test properly and carefully, the concordance is very high, above 99%. And in fact, I think ultimately they're going to replace the invasive tests. It's just a question of people getting comfortable. Because amniocentesis isn't perfect either. It's the gold standard, but they have a false positive, false negative rate, which are a little under 1%. So it's there's a number to beat, and I think as the numbers increase, the data will be illuminating, and I'm thinking it's going to look pretty good for these non-invasive tests. All right. So I wasn't involved in any of those clinical trials or anything like that, and while they were off doing that, we started thinking about other ways to use this counting principle, which is this idea of looking for overrepresentation of a particular chromosome, or a piece of a chromosome, or even just a particular location in the genome. And we eventually figured out that we could use this counting principle to measure the whole genome of the baby, not just whether there's an extra copy of a chromosome or not. And Wei Gu, who was a PhD MD student in my lab, did that. And we published it earlier this year. I'm not going to say much more about that except to say that it's sort of the logical conclusion of where you go when you're starting with chromosomes, and we managed to get all the way to looking at the genome, which has some clinical diagnostic value as well, I think. And maybe to give you a sense of how serendipity works in science and chance encounters, after we published the first paper on the Down syndrome diagnostic, word got around the hospital at Stanford I was interested in non-invasive diagnostics. And one day I got a call from a cardiologist named Hannah Valentine, and she said, "Hey, Steve, when we do heart transplants, we have this problem in that we worry that the recipient of the heart transplant, their body might reject the heart. And the way we handle that right now is after they have a new heart, we go in and we physically biopsy them. So this poor patients have gone through all the trouble of having a new heart put in, and then some doctor goes in every couple of months and rips out a piece of it so they can look at it under the microscope to figure out if it's being rejected because the patient's immune system is attacking it. That's not a good thing. And so there's been this very long search for ways to not have to do that, and she brought my attention to that, and we sat down, and I realized that all the stuff we've been doing on cell-free DNA could also be used in organ transplant patients. And so the idea there is to think about the heart transplant as a genome transplant. And here's the cartoon to have in your head. If your genome is red, all the parts of your genome that make it uniquely yours, the particular inheritance of polymorphism, single nucleotide polymorphism, if you heard about that, are red. All the molecules in your blood will be red. So before you get a heart transplant, the only DNA that's floating around in your blood is red. However, when that heart gets put in your body, that heart came from a different person. Every cell in that heart has a different genome. It has the genome of the donor. And so when any cells from the heart die, they're putting in, let's call it green genome, they're putting in green molecules into your blood. Okay? And so now, if you're analyzing the sequences of the molecules floating around in the blood, the red ones come from other organs, from the recipient. The green ones specifically come from the heart. And if the immune system of this recipient starts to attack this new organ and recognize it as foreign, more cells are going to die, and then more green DNA is going to come in the blood. And so this provides you with a really neat and effective way to potentially monitor the health of the heart. You sequence the DNA in the blood, and you identify which genome does it come from, or which portion comes from this foreign genome. And we did it sort of in a very similar way to the aneuploidy test, where we take some blood from the patients, we purify the cell-free DNA, and we sequence it. In this case, however, we're not looking for the universal aspects that identify which chromosome the molecules come from. We're looking for the specific parts that are unique to the donor and the recipient genomes. And we've measured those ahead of time, so we know the big parts of the genome of the donor and the recipient, so we have a table. We just look up where the molecules come from. And that's kind of shown here, where these are 5 different patients who received heart transplants. This axis is time in months after transplant. This axis is the fraction of donor or foreign DNA in the blood. And at every single time point here, these patients were physically biopsied. And a pathologist looked and said, they're rejecting the heart or they're not rejecting the heart. And the places where the pathologist said, hey, they're rejecting the heart, are all shown here with red arrows. Okay? And what becomes pretty evident is that, look at the blue line. The green ones have to do with the measurement noise. Don't worry about that. The blue ones are the actual donor fraction. You can see that the foreign DNA in the blood is peaking up every time the pathologist thinks there's a rejection. And in fact, it's anticipating the rejection. It's starting to go up before the pathologist sees anything. And in the case where there's no rejection called by the pathologist, there's clean signal there. This is Tom Snyder, who's the postdoc in my lab who did the work. This is Hannah, who I mentioned earlier, our collaborator. So that got us very excited. We published sort of the proof of principle there and then went out and got a grant to do a large-scale clinical trial. And this, in fact, was money that was available through the stimulus package that the Obama administration put through, the ARA Act. And with the supplemental money they gave the NIH, we were able to apply for a grant and do a sort of large-scale study, which is just concluding now. And so we've got something like 700 plasma samples from 100 different patients. We've done heart and lung. And so lung is a new one, and we can see that the signal is peaking up where there's rejection events. That's good. In heart, don't worry about this. This is something that measures the effectiveness of the diagnostic, called a receiver-operator curve, where you compare the false positive to the true positive rates, and it ends up being a pretty good diagnostic. And we're pretty happy about that. So now we're trying to figure out how to get that into the clinic. So let me change gears now away from cell-free DNA and talk a little bit about the immune system and ways to sort of sequence the immune system. I talked about how the immune system might, if you put an organ transplant in your body, you've got to worry the immune system is going to attack it. It recognizes the organ as foreign, coming from a different person. Those patients are actually immunosuppressed. They're given drugs to turn down their immune system. And the immune system is kind of an amazing part of our bodies in that it creates antibodies. So there's a part called the adaptive immune system that makes antibodies that will have an incredible ability to recognize foreign invaders, whether it's a virus, bacteria, or other sort of thing you're being challenged with. You can create a molecule that will bind it and say, get it out of here. And the antibody is the molecule that does that. So when you get vaccinated, and I hope you were all vaccinated as a kid, those vaccines are meant to create antibodies that protect you against all kinds of disease, tetanus, diphtheria, you name it. They're training your immune system and doing it in a way that gives you antibodies that are useful in case you ever encounter the real pathogen. And so this is what the structure of an antibody looks like. It's a protein. The business end is circled in green here. These are two business ends. That's the part that does the molecular recognition. And it's kind of amazing. You can ask yourself, how could I create a molecule that can recognize arbitrarily other shapes? And through many decades of research, biologists have figured out some important aspects of how this works. And combinatorics plays a big role. What happens is there's a selection of gene fragments that can sort of be cut and pasted inside B cells, which are the ones that make antibodies. And so it's shown schematically here. There's a bunch of blue fragments, a bunch of orange ones, a bunch of pink ones. And every antibody gets one blue, one orange, one pink that are basically picked randomly and then glued together. Okay? They're also given names, V, D, and J. They're glued together. And that combinatorics gives you a certain amount of diversity. And if you look at the total number of each of these segments and multiply them all out, it ends up being about 10,000 different things you can make. That's not a lot of potential antibodies. And so there are other ways to create diversity. One way that happens is that the gluing together of these pieces is not done very cleanly. So the cells can nibble away at the sequences or add random bases to them when they glue them together, and that creates a further level of combinatoric diversity. And then another important one is what's called hypermutation, which after the gluing together happens, the antibody gene can just be randomly mutated. So it creates cells will create random mutations, and they keep the antibodies that work best. The ones that don't work well get disposed of by the body through a complicated process I don't want to get into. And when you start considering all those processes together and say how much diversity could you have in your antibody repertoire, you get pretty enormous numbers. And so these are the V's, D's, and J's. This is human. Let's get human. Multiply them together, you get 10,000. Then you incorporate those other aspects, and you get something like 10 to the 14th. That's an astronomically huge number. And you can ask, do I have 10 to the 14th different antibodies in my body? And the answer is no, because the number of B cells you have in your body, which are the ones that make antibodies, is only about 10 to the 10th. And the way this whole thing works about that cutting and pasting of the genome, each B cell commits to making one and only one antibody. Okay? They have such a strong commitment that they've reprogram their genome in the way I just described to make that antibody, and there's no going back. Kind of like Cortez burning his ships when he got to the New World. Right? And these B cells, there's no going back to what they were. They picked an antibody. That's also apocryphal, by the way. He didn't burn them, he sank them. So don't, anyways. So 10 to the 10th is the maximum number. But people sort of had no idea how many there were. And you go through the literature and you ask a very sort of basic question of how many antibodies are in your body at any given time? What's the frequency and multiplicity, the distribution of them? Really wasn't known, and the estimates were all over the place. And so we got interested in trying to measure that using these sequencing technologies and to answer this sort of very elementary and fundamental question about the immune system. And so this is the work of a couple of really talented folks in my lab, Josh Weinstein and Jenny Zhang. Jenny's now a professor at UT Austin, Josh's postdoc at MIT, and collaboration with Daniel Fisher, who's a theoretical physicist. So I had mentioned there's something like 10 to the 10th B cells in the human body. That's a pretty big number. And if you wanted to measure all of them, it's sort of beyond the capability of current sequencing technologies. Plus, you'd have to exsanguinate your patient, take all their blood out, and that wouldn't be good. So we began looking for a model system. In biology, this is something that's a great tradition of that. Hydrogen is a model system in physics. Biology has many model systems, E. coli, Drosophila the fly, and so forth. And we stumbled on zebrafish as a great model for immunology. It had been used as a model system for developmental biology for many years. Turns out it's phenomenal for immunology. Because if you look back the evolutionary tree, look back 300-400 million years, that's when the adaptive immune system first arose, and it sort of first happened when fish got jaws. Okay? So all these antibodies, all that, they first were invented when fish got jaws. Somehow they started eating things that required them to have a more sophisticated sense of self and non-self. And it turns out that sharks, they have an immune system, an adaptive immune system, but zebrafish, they have all those same methods of generating diversity that I just described for humans. They do this combinatorial rearrangement of the junctions, they create diversity at the sequence junctions, and they do that hypermutation. And so it's a very nice model system because all that ways to make diversity are there, but there are many fewer B cells, something like only 10 to the 5th. So 5 orders of magnitude fewer cells to measure. And you can do experiments like this. Or you put the fish in a blender, you grind it all up, you don't care where the antibody-making cells are, you get the whole thing, purify RNA out. So Bassomatic, you guys ever watch Saturday Night Live? No, all right, one piece of homework for the lecture is go to Hulu or some other website and look up Bassomatic. You'll find an old Saturday Night Live sketch with a very young Dan Aykroyd. It's one of those cases where truth is stranger than fiction, actually. I started Googling around, and turns out liquidizing goldfish is not a crime. Some artist in Copenhagen had created an exhibit where he made basically a fish tank out of a blender and had it plugged in and was kind of daring people to go and push the button or come to the museum. So of course some people pushed the button and ground up the fish. And what happened is the guy who ran the museum got arrested for cruelty to animals. And I'm not making this up. This is on BBC, all right? And at his trial, they got a witness from the maker of the blender, Moulinex, who testified that the fish had died painlessly within 1 second of the blender being turned on. So they let the guy off. So, it's... check out Bassomatic. So what we do is we grind the fish up, purify out the RNA, which is the message for making the antibodies, and amplify it up, and then sequence it. And we use a sequencer to let us read the whole length of that heavy chain, the VDJ. And then you get all these sequences, you put them in the computer, and you get to play all kinds of games with them. And in fact, you can start doing essentially statistical physics on all these sequences from the immune system and ask all kinds of questions about what's the diversity, what's the mutation rate, how many antibodies are there, so forth and so on. So we had quite a bit of fun exploring that. It turns out that each fish looks quite different from each other one. If you look at how are the VDJs used, those different segments, how have they been recombined? And so these are 4 different fish. This is the V-axis, this is the D-axis, this is the J-axis. So each point in 3 space is one possible combination of those VDJs, and the size of the ball and the color is how many antibodies use that combination in this particular fish. And you can see that they look quite different. All right, so what do you do about that? You know, it's one of the complaints physicists always have of biology. It's not repeatable, everything is different. Well, you look at it a certain way, they all look the same. So if you just plot the frequency distribution, you don't care which one is the most frequent, but you ask what's the relationship of the frequencies. Turns out each color is a different individual fish. They all lay on top of each other in the universal curve. That's pretty cool. Same thing is true when you look at very, very high resolution. Looking at individual antibody sequences, taking into account hypermutation, they all lay on a universal curve. It appears to be a power law. Physicists get excited about that because they understand the sort of basic mechanisms that give rise to that sort of shape of distribution. You can estimate the total number of different antibodies, which was kind of one of the first questions I asked. And there's different ways to estimate it, but it turns out a few thousand different antibodies in each fish. I think that's all I'm going to say about that. Except if you want to get a sense for how these measurement technologies are changing, the amount of data you have in biology, this is the total cumulative number of antibody sequences that have been measured in the history of the universe, which started appreciably in 1993 and increased up to this number. This is times 10 to the fourth, so about 80,000 by 2008. One paper that we published looking at these fish changed the curve to make it look like that. Right? That was one paper. And this is kind of amazing. And we're in a really amazing time in science where we have this ability to generate all this data, ask all these questions, and really look at different aspects of biological systems from a completely different direction. And that's a lot of fun to play with. And it's great news if you're a zebrafish. We've gone on and done quite a bit in humans as well, but I'm not going to tell you about that today. What time am I supposed to finish up?
I think you started about 10 or 15 after.
Okay.
So once you go to 10 or 15 after.
All right, I can do that. Dark matter. How many people have heard of dark matter? Yeah, all right, in physics, right? I mean, that dates back to the 30s, but, it's sort of folks like Fritz Zwicky and Vera Rubin were looking at astronomical effects where they could see that how galaxies rotated, try to apply Newton's laws to angular momentum and whatnot, and it was completely inconsistent with the amount of mass that was observed to be there through telescopes. So, oh, there's a problem. There must be some dark matter out there in the universe. And been verified other ways by looking through the Hubble Space Telescope. You can see these distortions are caused by gravitational lensing. And so in physics, there's this huge problem that, in different ways you look out at the universe, you get different answers about how much matter is there. And the vast majority of what we think is out there is invisible. So you can have dark energy, dark matter. This is sort of the stuff you can see. And all the rest is stuff that's inferred through sort of indirect measurements, but is an inconsistency with what you can see. That's a huge problem. It's not my problem, thank goodness. But biology has a similar problem. It's a dark matter problem in biology, which is that people can sort of make indirect measurements of what's out there, especially in the microbial universe, and then they look at what they can grow up in culture, which is sort of the tool for microbiology. And 99% of what's out there can't be grown up in culture. And so there's a huge amount of biological dark matter out there too, whose existence has been sort of studied indirectly, and I'll give you an example of how that's done. So this is the tree of life right here. Then you may say, where does the tree come from? So there is a gene that is sort of ubiquitous. There's many genes that are ubiquitous among all living creatures. There's one that people have focused on in particular because for technical reasons it's easy to measure. And so people measure the sequence of that gene in lots of different organisms. Then they compare the sequences and look at which ones are closest to the next. Can you get there by making very small numbers of mutations, or do you need lots of mutations? And that's what this tree tells you, effectively. How many changes do you need to make to this gene to get from one organism to another? And if you felt you were special at all, let me disabuse you of that notion. Okay, here's animals. This branch right here is animals. And it is stuck right in between slime molds and fungi. Okay, so in sort of the diversity of life on Earth, slime molds, us, and fungi are this little bit right here. And then there's everything else. Okay, and all of this stuff over here are all essentially single-celled microorganisms, bacteria and archaea. So the vast majority of evolution, diversity, and life are these microorganisms, these things that are dark matter that can't be grown up. Everything you see with your eyes is essentially these four branches right here: molds, animals, fungus, plants. Okay? So most of the world is microscopic, and the breadth is enormous. I mean, it's really incredible. And so we got interested in this, and I can tell you sort of another way to think about the problem. is you guys all learned the Linnaean classification system, kingdom, phylum. King Philip came over from Great Spain, genus, species, all that, right? So phylum is kind of the second grossest way of distinguishing species. Kingdom is the grossest. Phylum is the second grossest. And these are bacterial phylum, shown here. And all the gray ones are the ones where there's never been any cultured members of that phylum. You couldn't grow them up and study them in a Petri dish or a test tube. And see, there's a lot of them at the very grossest level of classification. And we began to get interested in trying to solve this problem using some of the measurement techniques we developed oriented around these microfluidic devices. And I'm going to tell you a story about TM7. Well, one of the ones I'll tell you about is TM7, which you'll note is stuck in this desert of phyla where nothing around it. There's no genetic knowledge at all. All that's known is the sequence of this 116S gene. And that was used to make these trees. So microfluidics, I mentioned them earlier. It's kind of a way to think about making automated plumbing. This is a picture of a chip we made in my lab. It's all been filled with food coloring, so you can get an idea of sort of the complexity of the plumbing. The channels here are about the width of a human hair. And this chip has, I forget, 600-700 valves on it. And you can see that it's starting to kind of look like an integrated circuit. And we spent a decade in my group developing the ways to make these chips and use them for all kinds of different problems in biology. But in particular, one thing we're very focused on right now is using them to do single-cell genomics. So combining these tools with the sequencing tools I've been telling you about to ask questions about individual cells. And this biological dark matter is particularly interesting to us. And we've spent quite a bit of time developing different chips. This is one of them that allow us to isolate individual bacteria, amplify their genomes on the chip, and recover the material and then sequence it. And TM7 that I mentioned was one of the first ones we worked on. We've had a bunch of great collaborators from the microbiology community. So TM7 was a new phylum, OP11 new phylum, OP9 new phylum. I sent one of my postdocs out to Yellowstone Park. We went camping for a week and sampled from the hot springs there to get some of the samples we looked at. This one is interesting. It's the world's fastest swimming microbe. It swims some hundreds of microns per second, and they weren't able to culture it, but in a course at Stanford, the students isolated it, amplified it, and we sequenced the genome. This one right here lives in the San Francisco Bay. It's an archaea, and it's involved in processing ammonia. These types of bugs, when they get out of balance in the ecosystem, you get red tides and things like that. And so they play a very important part in maintaining ammonia balance in the oceans. And so there's many fun stories for each of these. And all these papers are up on my website. If you're interested in that, you can download them and read them. I'm going to take you to the next level of complexity in single-cell genomes, though, which is looking at human genomes. So I talked a bit about personal genomes. Personal genomes are actually a bit of an average because it's not the case that each of your cells has exactly the same genome. And that's true for a bunch of different reasons. One that we've talked about is the immune system. Those B cells that make the antibodies, they've all reprogrammed their genomes so they have a different gene at that antibody location. And they're all distinct. And so if you look at someone's personal genome and try and look at that particular gene, it's a mess because every cell is different. And people just kind of ignore that, which is fine. Gametes, all right, sperm, egg cells, those are all different. For one thing, they're not diploid, they're haploid, right? Because you mate with someone, you give half of the genome to the child, they give the other half. So you've got to have a way of giving only half your genome. And that's one reason they're different. Another reason they're different is that they're kind of a mixture of your inherited genomes. And I'll talk about that in a few minutes, specifically how that works. But another example is cancer and aging. Okay, so the way cancer happens is when cells divide, they don't create perfect copies of the genome. They make mistakes. Those mistakes are mutations. If they happen in important genes, and you mutate an important gene, one that sort of is involved in cellular self-control, your cell may lose its sense of self-control, start to divide without limit. That creates a tumor. The tumor has a distinct genome from the rest of the body. And many of the cells, as you age, all your cells will have slightly different genomes. And so trying to understand cancer is intimately entwined with trying to understand how mistakes get made when cells divide and how genomes change from one cell to another. If there's time at the end, I'll give you an example of that as well. But let's go back to the gametes. This is pretty fun. They still do sex ed in high school, or is that all forbidden now? They do that? I see a few nods. All right. So you can, as we got into this, I like to go back to the earliest literature, right? That's what scholarship is supposed to be about. So it turns out the ancient Greeks, back in antiquity, they knew something about sex and reproduction. Actually, it's frightening what they knew. So Anaxagoras, he had worked out that the male determines the sex of the baby. He was right about that. All right? The sperm determines the sex of the baby. When there's an X or Y chromosome, women's are XX, men are XY. Men determine the sex of the baby. However, he thought the way it worked was that the male seed came from the right testicle, the female seed from the left. So this question of that turns out to be not the case. They did experiments to test that, however. They used to try to use a ligature to tie off one testicle. They were bulls. So this is with bulls. I haven't found the official publication, but it's been cited in a secondary way. Probably didn't work the way they thought. You can check their statistics. But this idea of asymmetry was very important in the ancient Greeks. Okay, they had mathematical models of everything. Mathematical models of development, ideas of symmetry. Symmetry is very important in modern mathematics and physics. So there's kind of a nice connection there. And their ideas, the Greeks' ideas of symmetry, extended to all kinds of things. They thought milk from the right breast was stronger and more suited to male infants. Diseases of the right side were more severe. All that, of course, we know now is not true. But that's how science works. You make a hypothesis, you test it. Aristotle had general biological theory of left and right. And so that leads you to this question of, how much of that was right, how much was BS? Is there really some left-right asymmetry in the male gonads? Turns out it is an anatomical fact that the right testicle tends to be higher and heavier than the left, and there is an asymmetry there. And this was worked out quite rigorously by these pathologists back in 1960 who had nothing better to do, and they were going to take cadavers and cut them up and weigh the testicles and measure them. All right? It's kind of amazing. The Greeks, they did know something. And the Greeks knew it. How do you know the Greeks knew it? All right? This is pretty interesting. How do you know the ancient Greeks knew that? Well, some very enterprising young PhD student back in the 70s went around the British Museum and he looked at all the statues from antiquity and he measured the testicle sizes. And he plotted up the height asymmetry, the size asymmetry, and the percent of statues with testicles the same size. He published this in the most prestigious journal in science, which is Nature, and he won an Ig Nobel Prize in 2002 for his work there. 1976 turns out to be a very silly year in the history of Nature. There's a whole series of articles that follow this up, and I won't bore you with that. But remember our friend Anaxagoras, right? So he is right here, right when the Greeks were figuring out that the knowledge is making its way through the artistic community. That this is how you should represent human physiology. Pretty amazing. So, we're interested in ideas of asymmetry, but now we live in the molecular age. Okay, so we're interested in asymmetries of the genome in ways that not just physiologically different, but in the way the molecular genetics is different. And so we got interested in trying to measure this in the way that recombination happens. How to say this? Your genome, you inherited half from your mom, half from your dad. And when you make a sperm or an egg cell, that's a haploid cell, so it's got half your genome in it. Which half goes into the sperm and the egg? The mom's half or the dad's half? It's a mixture, right? So it turns out through this process of meiosis, you've learned about mitosis, which is how cells divide when you're growing. Meiosis is how germ cells divide. During meiosis, you mix together the two genomes in random ways and create haploid genomes that are more or less half one, half the other. And we got interested in trying to study that sort of recombination. Let me show you how it works. Here imagine you've got a genome that's got two chromosomes. It's diploid, so you got the two blue chromosomes from your dad, two red ones from your mom. Through meiosis, these chromosomes associate with each other and they can cross over. So physically, bits of it where they touch can cross over. They get cut and pasted, recombined. You create these mosaic chromosomes, and then as the cells divide, they segregate. You create these haploid daughter cells. Remember, the original was diploid, 2 copies of each chromosome. Sorry, 2 there, 2 there. The daughter cells have 1 copy of each chromosome, and rather than just picking the maternal or paternal inheritance, they've cut and pasted things together to create these random procedures. This process is called recombination, and it's an important part of human genetic diversity. And there have been a number of studies, large studies across all of human diversity, different sort of human population and cultures across the world to understand exactly how recombination has happened through the history of the human race. And those have been provide a very powerful insight into the history of humanity. We got interested in asking the question, how does this recombination vary within one individual? So if you look at a bunch of different sperm cells from one male, for example, how much diversity is there in the recombination? Does it reflect what's happening at the population level, or is the population made up of people with much different recombination activities? So Jianbin Wang in the lab led an experiment to do that. And the experiment, sample collection, nothing very high-tech about that. And was very pleased that for the first time in my life, I was able to write a scientific paper with the word masturbation in it. We took the sperms that were collected and put them in one of our little chips where we could isolate the sperms individually, separate them out, amplify their genomes, and recover the amplified material from the chip. We then sequenced it, genotyped it, and did all sorts of analyses. One of the sort of important parts of this was to take each single sperm genome and map it back to the personal genome of the sperm donor. And we had haplotyped that person's genome, so we knew which of their chromosomes came from their dad, which from their mom. And for each single sperm cell, by looking at which mutations or SNPs were associated with the maternal and paternal chromosome, we could work out exactly what recombination events had created that particular sperm cell. And so for each cell, we're able to build a map like this. So this is kind of a synthetic karyotype, where for chromosome 1, and we're haploid now because it's a sperm, we could see that there was mostly, let's call it paternal contribution, but this part of chromosome 1 came from the maternal half of the genome. So there was a crossover event right there. Chromosome 2, there were several crossover events, and so forth and so on. So we collected this sort of statistics for each chromosome. And we can compare them to the population-wide estimates across all of human diversity and say, those folks have measured. What's the probability of recombination happening along these are all the chromosomes here, so along a chromosome, this is kind of a cumulative distribution of probability. It's more likely you get recombination happening at the ends than at the middle. The lines are their data, the dots are our data, and they all more or less lie on top of each other. And so you get just as much sort of recombination diversity within one member of humanity than you do across the whole human race. Really kind of amazing. And for those of you like physics, I call this an ergodic principle of recombination. We took a few of the sperm cells and sequenced very deeply into them. So we effectively did the whole genome. And we were able to sort of measure what was the mutation rate in the sperm. Okay? All these things out here are places where that sperm cell had complete disagreement with the donor's reference genome, and they're therefore novel mutations. The mutation rate, we had a bunch of arguments about the actual number with the referees. Turns out our number was right, and we're very happy about that. There's another small lesson in science there, but I won't get into that too much now, because I want to say one more last sort of vignette, or a couple slides about cancer and how these single-cell approaches are helping us understand the origin of cancer. This is work we did with an oncologist, Ravi Majeti at Stanford, my postdoc, Tom Snyder, again, his student, Max Jan. And we got interested in trying to actually measure the mutations that cause cancer. That's actually turns out to be a really hard thing to do. Everyone's got this picture in their head. You get a couple of key mutations and these key genes, and all of a sudden the cancer takes off. That's called the multiple hit hypothesis. And it's never actually been observed directly in a human cancer. Okay? It's been inferred indirectly. So you can infer it a couple of different ways. One way is by looking at the incidence curves of cancer, look at the rate at which people get cancer as a function of age, and it turns out the shape of that curve is consistent with there needing to be multiple events happening, and that was kind of the original way of doing it. It's called epidemiology. And then people have done model systems in organisms like mice. Okay? So they'll physically, they will go in and sort of in a targeted way, mutate a gene that's known to be related to cancer, and then they can cause cancer in the mouse. And they can show that the mouse needs to have multiple hits in order to get the disease. But that doesn't necessarily mean that's the way it works in humans. We got interested in trying to measure it directly in real human tumors. And so we focused on leukemia, which is the cancer of the blood, for a variety of reasons, technical ones. We sequenced the tumor, we sequenced the normal genome of the person, we found mutations that were specific to the tumor. Then we went back to some of the remaining normal cells in the blood and said, do these normal cells have some of the tumor mutations? In other words, were they early steps on the way to making the tumor? And it turns out we could see that some of the mutations were there in the normal cells. All right, oh, that's good. Then we went and we took a bunch of these normal cells and did single cell genotyping of them to ask, in what order do those mutations occur? So here's mutations from the tumor. They occur in some of these normal cells, but what's the pattern of occurrence? And it turns out that there's a very sort of defined way in which, in relationship in the occurrence of these mutations. And so these are genes on these axes. Each blob is a single cell. And you can see that most of the cells for this patient didn't have any of these mutations. 50 of them had this one mutation. 3 of them had this mutation and these 2. And 1 of them had all 3. And it never occurred any other way. Not in this patient, not in any other patient we looked at. It's always the case that there's a very direct sequence of mutations. There's a sort of Venn diagram that's perfect concentric circles among these cells. And so we're able to say, sort of definitively, this was the order of which these mutations occurred on the path to cancer. And these happened to occur in the normal cells before there was disease, or the earliest, probably the most causal of the mutations. And in this way, we're trying to sort of dissect this very complex and heterogeneous disease, and this combination of single-cell manipulation techniques afforded by microfluidics and high-throughput sequencing have enabled us to do that. And so, I'll come back to the slide, which I showed you earlier on. These measurement techniques are things that are really changing biology in a way that's quite powerful. And so I've given you examples of non-invasive diagnostics for Down syndrome, organ transplant recipient diagnostics, ways to measure the immune system, and finally, ways to look at human reproduction and cancer. Just in case it all sounds so good, let me give you a last word of caution. Maurice Wilkins is one of the guys who won the Nobel Prize for discovering the structure of DNA, and he was a little bit disenchanted at the end. It's his Midas gold. Everyone who touches it goes mad. So I'll stop here and take any questions.
Have questions?
A greater amount of the chromosome 21 in a fetus. What could be done about it? What would be the benefits of protecting it?
The question is, how do you detect the extra copies of chromosome 21? What's the benefit of detecting it early? Yeah, that's a good question. So why bother doing this diagnostic test at all before the baby's born? Well, turns out many of the parents who discover they've got a kid with Down syndrome, they're going to terminate it. In the cases where they don't want to terminate, they decide they're going to have the kid regardless of genetic disease or not. Turns out there have been studies that show the earlier the parents know, and the better prepared they are to raise a kid with special needs, the better the outcomes are for both the kid and the parents. And so sort of independent of what the decision is to do with the knowledge, it's better to have the knowledge, is what the studies are showing.
Yes. No, you.
What if you were to do the test like a day after the child was born? Would that lead to the same kind of benefit for the child as no?
Yeah, that's a good question. So why not wait till the kid's born to do the test? There are examples of that are less charged than ones arranged around termination. So let's talk about things like metabolic disorders. Maple syrup syndrome, PKU, where these are genetic diseases where kids can't metabolize certain amino acids, and so they are essentially cured if they eat the right food, and they get sick if they eat the wrong food. What happens today is the kids are tested right when they're born. There's a little heel prick that's done, and you get the results back in like a week or two. All that time, the kids are getting sick because they're eating the wrong stuff. It's like mayhem in the hospital. And so the benefit for conditions like that of doing a prenatal test is you'll treat the baby right when they're born, and they'll never be given the stuff that makes them feel sick. So that's, I think, part of the value of doing the genome. That's not an aneuploidy, those are point mutations, but that's the sort of information you get out of doing the genomic the genome measurement.
In the back row.
For the microfluidics chips, those help you actually culture the bacteria that weren't even able to be cultured before, or simply isolate the genome in a smaller number?
So you're asking if we do microculture on the chips, or yeah, there are people who've been interested in that. It's not what we've done. We've more just been trying to use them to isolate the organisms and then amplify the DNA directly.
Is that at some point in the future you'll be able to like shut off the genes that express the mutations for cancer, and that way clearly preventing person from developing cancer later on?
Yeah, so so the question was, can you turn off the genes, the mutated genes that are causing problems in cancer? And that's a very active area of pharmaceutical research right now. People are developing what's called targeted therapies, which are drugs aimed at disabling common mutations in cancer. And so Gleevec, which is a famous one in leukemia, is a targeted therapy that's aimed at one particular mutation that happens in some fraction of leukemia patients. And these are very promising. Each one only addresses a segment of people with a given disease, but the thought is you get enough of them, use them in the right combinations, it'll be a very powerful approach to treating cancer.
In the event that someone were to receive a transplant from a close relative or presumably a twin, would you have enough resolution to determine between their DNA?
That's a good question. So if you get an organ from a family member, can you resolve the DNA? And the answer is, as long as they're not a twin, you can do it. Yeah. That matters mostly for things like kidney transplants and a little bit for liver. But for heart and lung, there's not enough organs. They don't even bother trying to find family. Well, family members are not going to give you a heart anyways. But there's just not enough organs that they can do any kind of even HLA matching. So they just stick whatever's available and whoever needs it.
For the transplants, you look at foreign DNA that's in the blood, the scraps, to see if they're going to reject it.
Yeah.
Is there a way to do like a test when you're young where they take blood, look at the scraps of DNA, and each year you take another blood sample to see, look for scraps of cancer DNA? Or is there too much stuff going on during your lifetime that you get all kind of background noise anyway?
So you're asking, can you use this cell-free DNA as a way to do a cancer diagnostic to look for mutation? Yeah, so there's a literature on that that's gone back for a couple of decades, and clearly it works in principle, and there's been beautiful papers written on it. It's never made it into the clinic as a practical diagnostic for reasons that are not really clear to me. And my sense is that people are about to take another run at it thanks to these sequencing technologies, and that may be the thing that helps get that approach over the edge. Yeah.
Against the wall, yeah. No, you. In the check shirt.
Sorry. On the discussion about family members and rejection, is it possible for you to reject a twin's organ?
Well, it depends whether they're identical or fraternal. Yeah. The theory is that's a perfect match. I suspect it's probably pretty good. There must be literature on that for bone marrow transplants. I actually don't know what the answer is, but someone must have done bone marrow transplant from twin siblings. I'll bet you it's pretty good. There are cases of, I'm trying to think now. I did look at this at one point for another question. It wasn't between twins, but people have allergies. So people have gotten bone marrow transplant is basically they kill your immune system and give you someone else's. So it's like a transplant in the immune system. And people who haven't had allergies, they get a transplant. People who have had allergies, they get the allergies and vice versa. So allergies come along with the immune system.
My brother just had he had a bone marrow transplant. He was highly allergic to cats and dogs. He got it from my brother, and he came to my house, and nothing. He was fine.
There you go.
Anything, yeah.
There you go.
Amazing.
You said that some or lots of phylums of microbes can't map their genome. And why is that exactly?
They can't be grown in culture. Okay, so the tool for microbiology is you take a bug, you try to isolate it, you grow it up. This goes back to like Koch's postules for disease, but it's also more generally done in microbiology. If you can't grow it up, how are you going to study it? The vast majority of those can't be grown up.
When you were looking at the differences in the proportion of DNA in the blood to determine if the transplant was being rejected or not, did you find or did you fear that DNA from symbiotic bacteria might influence that? So, like, are there bacteria in our gut, throwing DNA in our blood also, or things like that?
Right, so the question is, do we see microbial DNA, bacterial, viral sequences in the blood when we do the transplant measurements? Well, that touches on a lot of interesting things. The short answer is yes, and the longer answer is, there's something called the human microbiome, which is to say you are colonized by bacteria. And in fact, if there was a vote, if it was a democratic process, every cell was allowed to vote once, you have 10 times more bacteria in you than you have human cells. So you are, by sort of a number census, more bacterial than you are human. It's kind of amazing. And so when we sequence the cell-free DNA, about 99% of what we sequence ends up being human, about 1% is microbial, which is exactly what you'd expect, it turns out. Because if you think about the fact that the human genome is 100 times bigger than most microbial genome, it all kind of works out. Or 1,000 times bigger. So we do see it is there at a low level. It's really quite interesting. And we're just in the process of publishing a paper now looking at the viral load in people who receive these transplants, because they're a very interesting cohort because their immune systems have all been turned down with drugs, right? They're all immunosuppressed so they don't reject the organ, but that also makes them more susceptible to viral infections. And we can see the whole viral distribution changing as a function of how much drug they're being given.
Do you see your technology maybe being used to diagnose disease in very unclear cases, identify exactly which pathogen is there?
Yeah, so the question is, can you use this sequencing stuff to identify pathogens and do molecular diagnostics? The answer is yes. There's a bunch of examples in literature, people who've done that. People get an infection. Doctors don't know what it is, don't know how to treat it, trying to figure out. You can sequence some of the material and sometimes discover the virus that's causing it and what sort of virus it is and how you might go about doing it. It's done not that frequently now, but there are some proof of principle examples out there.
How large are those chips that you were showing us before? Like, do you think that maybe in the future you could have them integrated into like a finger prick device where you can just go to your doctor's office and get your DNA sequenced?
Yeah, the question is how big are the chips? They're about 1 inch, so yeah, they're very sort of portable sort of thing, quite small.
Do you think that's feasible? Could that actually happen?
Yeah, so there's been huge sort of academic literature on this idea of making point-of-care devices that would be sort of doctor's office type things. You go in, finger prick, whatever, and there's no technological reason why that's not happening right now. All the technology has been demonstrated, and I think it's purely a question of economics and the realities of how healthcare works today.
You said that fetal DNA is in the mother. Is there also transfer to the mother when it's the fetus? Is there also internal DNA transferred to the fetus?
Oh, does the baby see the mother's DNA? That's an interesting question because the same blood circulates, and certainly through the placenta there might be some transfer. I don't know if anyone's looked for it. I wouldn't be surprised if there's bits of it there.
When you see people always talk about, especially in a more science fiction way, like Gattaca and stuff about how you in the future maybe analyzing your DNA for certain propensities towards certain conditions will be able to predict that very well. How we talk about I read things today about you can detect genes that do indicate some propensity towards being vulnerable to certain diseases, things like that. But do you know how actually practical to predict that is, or if it's extremely unreliable?
So you're asking, what do you really get out of sequencing a personal genome, and is it useful?
It's feasible in the near future to actually be able to predict with very concrete, or not very concrete, but reasonable certainty that you have a high risk.
Yeah, so it very much depends on the condition. There's a big spectrum. So the question is, again, how reliable are these predictions you get out of a genome? Some things are, fairly weak predictions. Your risk of some certain disease goes up by a factor of 2, and so all right, that's big error bars, epidemiology. And other things are very concrete. Some of the mutations in BRCA1 have, very enormous increases in risk of cancer. And so there's a whole spectrum of things. It's the gamut.
Do you think something along the lines of, you probably know the movie Gattaca, right? Yeah. So do you think something along the lines of that is entirely science fiction, or do you think that that's like, somewhere in the middle, reality?
The eugenics part of it?
Yes.
Yeah, well, we've been through that in the 20th century already. So, people have tried to do with cruder tools. It's, I'm not going to say it's not without possibility. In this country, the Bush administration signed the Genetic Non-Discrimination Act into law, which I think is hopefully an important step in preventing that from happening here. Yeah, so the question is, how do you turn off the diseases that were involved, the mutations involved in cancer? And so, it's not done in sort of peremptory way. The way it works is people wait until you've got the disease, they see if you've got the mutation, and then there are, typically small molecules, but sometimes also antibodies that have been designed to disable the protein after it's made. And so they compete in the binding spot, and that's how it works. The chips, what do we make them out of? Is that the question? Out of rubber. So it's a silicone elastomer, the kind of stuff you caulk your bathtub with. And we use a molding process. So we make the molds using lithographic techniques from the semiconductor industry, so sort of pattern transfer, photographic techniques. And then we mold the rubber.
One last question? If not, let's thank our speaker once again.